Of course, there's a lot of words that start with micro. No, it's not in here. So normal microbiota is in here. And microbiome, oh actually, no, yeah, it's definitely not in here. So the microbiome is just another way of saying normal microbiota. Where it comes from, the word itself, is that people always studied the normal microbiota either microscopically, so you could gram stain or whatever, but people, traditionally in microbiology, what you do is you go to an environment, in this case it could be our gut, it could be our skin, and you isolate organisms out of the environment. And isolation means that you take the organism and you typically get it onto a petri dish, the agar in a petri dish, as an isolated colony. And in laboratory we'll go through what it means to isolate a colony, doing quadrant streaking, whatever. But then you've got a colony that's in pure culture, and you can study that colony often biochemically, also microscopically, to get a good sense of what the organism is all about. And people would collect up lists of the kinds of organisms as well as their frequency in, for example, our gut or in our feces, which is, of course, easier to collect than material within our large intestine or small intestine. And the gut bacteria and fungi and everything else, and the skin on your eyes, in your mouth, together these represent what are known as our normal microbiota. They're the microorganisms that live in and on our bodies. They don't live in our tissues, ideally, but the gut itself is considered to be the outside of our body, and so is the lumen of the lungs. So, certainly by 20 years ago, people had already begun to make the shift over to, rather than studying microorganisms and distinguishing them based on their phenotype, what they look like, how they act, they started to study them based upon their genotype, which is the nucleotide sequence of their DNA, or in the case of some viruses, RNA. And then people came up with the idea that you didn't, well, first to sequence DNA you had to have a lot of DNA. And then we had the Human Genome Project, and we got very good at sequencing DNA. And so the amount of DNA that you actually needed to get a sequence of an organism became lower and lower, until now you really just need one piece of DNA. So you could have one bacterium with one chromosome in that bacterium. At least in theory, you could sequence all of its DNA. It's not necessarily so, because there will be mistakes, and it's better to have more than one sequence so you can figure out where the mistakes are. And the DNA might break or degrade, and so you might not get all of it. But anyway, in principle, you can get a sequence out of one piece of DNA. And so we call the study of the DNA, the genomes of organisms, genomics. And people started saying, well, we don't just need to isolate an organism before we sequence it. We could just isolate all of the DNA from an environment. And isolating all the DNA from an environment, where did I put my chalk? It's right
By your hand.
there. Thank you. That's bad. Yeah, now I can see it. They should make these a different color so it's not as similar. Or I should be using my colored chalk here. So they call the sequencing of an entire environment metagenomics. And the microbiome, the concept of it, although it's been broadened, was based upon the idea of making metagenomic studies of the organisms, such as in your gut or on your skin, so that you aren't just seeing and quantifying the organisms that you're able to get into pure culture, because you typically can't get all of the organisms into pure culture. If nothing else, there's typically a bias towards getting into pure culture the organisms that are aerobic, that can grow in the air. Because the anaerobic ones, especially the strict anaerobes, require that you remove all of the oxygen in order for them to grow, so they're hard to grow. So there's a great bias towards studying things like Escherichia coli, because Escherichia coli is trivial to culture. But with metagenomics, you don't have to culture. In fact, ideally, you don't culture the organisms before you isolate their DNA to sequence it. Now, there are a lot of biases in it, and you can intentionally put in biases. Like you could set it up so you're just looking at the viruses in the culture. Or you could just look at the free DNA in the culture. There was some study about how you could keep track of, I think it was killer whales in Puget Sound by taking samples of water and looking for the DNA that's found in the mucus that they shed from their bodies as they're swimming around. So there's many different ways of doing this, but people were able to get a snapshot of, in a sense, all of the organisms, or at least all of their DNA that is found within a given environment using this metagenomic approach. Takes lots of sequencing, but now we're pretty good at sequencing. Almost too good, I would say, but that'll be a different story. And so the idea of the microbiome initially came out of the idea of the metagenomics of our normal microbiota. But people like the word, I mean, this is literal, people like the word microbiome so much that they have come to use it to talk about normal microbiota, but also abnormal pathogens and things like that. Just what is there? We just call it our microbiome. We talk about harming our microbiome, for example, by taking broad spectrum antibiotics, which kill indiscriminately and thereby modify our microbiomes. We're actually, as a species, have this crisis in terms of our microbiomes, because as you take on this modern life where you're less exposed to the environment around you and you're taking antibiotics, maybe you're not breastfed, maybe you're delivered by C-section instead of vaginally, and we're gradually degrading our microbiomes, perhaps not gradually, perhaps faster than gradually. And we're finding out right now, because people are now doing these microbiome studies, correlates between what's in people's microbiomes and their degree of health. And they're finding all sorts of correlations between the kind of organisms that are there or absent and different kinds of diseases. One disease that is, or I don't know if you want to use the word disease, but one characteristic that people are associated with the microbiome is the amount of adipose tissue they have in their bodies, where they're finding that certain microbiomes seem to lead to a skinnier body type, and other collections of microorganisms associated with people are associated with a fatter body type. Another important thing is that the microbiota, the normal microbiota, is capable of microbial antagonism, which means that the mere presence of all of these organisms associated with our body makes it more difficult for pathogens to invade our body. Now, if you get a cut, you're in a sense bypassing your microbiota. But for an organism that is not gaining access to your body through means such as a cut, the organism first has to come in contact with you, then it has to in some way attach to your tissues, and then it has to grow into larger numbers, and maybe it becomes invasive, or maybe it makes a toxin that makes you sick. But in order to get that foothold, there has to be a room for that organism to, in fact, do that attaching and that growing. And if all of the spaces are taken up by your normal microbiota, if all of the nutrients that are available are being consumed by your normal microbiota, then these pathogens have less of a potential to get a foothold, and therefore less of a potential to cause disease. So this word, not last fall, but fall of 2017, when I last taught this course, it was like every other news item that I came across was about the microbiome and its importance in different aspects of health. All of those things, all of those posts are sitting there on my blog. You just have to go to the Facebook page and scroll down far enough, and you'll be able to see lots of things about microbiomes. And maybe I'll be finding a lot about microbiomes as well. In fact, one of the, this semester, one of the videos that I posted is basically a video on microbiomes. So just drawing to your attention that one of the things that's missing, that's a really important aspect of modern microbiology or medical microbiology, is this concept of microbiomes.